All right, our class is started. Welcome back from spring break, everybody. Oh my, let's see. Everything's been set up. I have a new computer. I apologize that, oh geez. Apologize that the recording on our last class meeting didn't work. That's always a possibility, and it's a frustrating one for everybody if it doesn't work, and it didn't work. And so we roll on from there. And that could happen again because I have a new computer. But hopefully it won't happen because I have a new computer. But I'm still getting used to my new computer and accessories and rolling forward. So this is where we're at. And we have these internships. If you're going to be around this coming school year, the application deadline is this Friday. The application is first sent to me. I just help get them organized and complete, and then send them on for review and interviews at MHR. And so because I'm in charge of it, and if you want to apply or if you have questions, ask me. And if you can't quite meet the deadline, let me know, because it takes them a while to get through the stuff. So it's really exciting, and once you've taken this course, you would be a star down there. There's this event also that's happening here in Portland. I think it's over on the University of New England campus. It's actually hosted and organized, sponsored by this BIO Association of Maine, and it's a free event. I think they always have good snacks and hors d'oeuvres kind of thing, so I would just go to this. And you can find it on a recording. There's the link. I think you probably have to register, okay? And so this is what we're up to. So we have our assignment due today. It's that CAR T-cell therapy assignment. And to fit that into our course as an example of cancer therapy that's covered in Chapter 19 briefly, but you know, it's really satisfying to see how a complicated modern attack by scientists and doctors on cancer can be explained in the context of our course, and how satisfying it is for how they built specialized human super receptor, and why they did it, and you know why it works, and the logic, the setup, the planning all comes from our course topic. So it's a good one, and that's due. There's some drawings to do on that. That's due for me today, and that of course will be included on our assignment. Our exam. And I also posted a video during break to cover the last parts of 19 that I really wanted to make sure we covered, so that you get those, you know, in plenty of time to start reviewing for the test. And we have our exam on Thursday, and it won't be surprising. It'll be like our first exam. So 50 short answer style questions, the same style as our first exam. And that'll be the same for all the exams in our course. The final exam will cover everything. Other than that, it looks like that. And, there's no tricks, no sneaky attacks on the exam. Any of the exams, it's just what's this, you know, how that makes that, explain that, draw that, label that. Make sure that you look at the wording on the exam. If it says draw and label, you got to draw and label it. Remember, you have to label genes really well to constantly demonstrate that you understand the organization of them. And that'd be true for all your drawings. They'd be simple, stick figure drawings. You don't have to be an artist. I'm not judging you on that. But, you know, drawing says a lot. And so if it says draw it, you got to draw it the way it describes. And if it doesn't say drawing, you can draw it. But if it says describe, you also have to use some narrative words, some sentences explaining what it is. But you can use a drawing to support that. You just need to explain, you know, some of what you're drawing. And then another thing to mention is, again, you know, you got to just keep working. It'll be that same thing where you're like, dang, you know, you just have to keep working through the exam to finish it on time. And we'll plug away at it like that. The folks who didn't do as well as they were on the first exam, well, we have a total of 4 exams. And each time the exam increases in its value, right? So the idea is, okay, so you got kind of caught off guard. You know, we tried to set it up with assignments and a couple of early quizzes and stuff, but you kind of got off guard with the first exam. Well, you can still get a fine grade. And that happens every time we teach this course, that, oh, wow, students are really surprised at the first exam of what really, that really is expected. Now you've seen that. And if you haven't looked at the first exam's video key, make sure you look through that, because I'm not, specifically asking you to go back and study that material, but for example, the stem cells that we were in the muscle, they're included on this exam, you know, so I would think that you'd go back to some of that material and the MIC and things like that, just because it's relevant for the context in which I'll be asking questions for this exam. And then it's just good to see that format and that style and understand, you know, the kind of style of answers. They're short, they're direct, you can use more words, but it's good as you're studying to try to find out how would you answer this. And you're welcome to, you know, still send me questions, issues, stuff like that for the exam. We can still time to meet, things like that. So that's where we're at. And our big focus today is we can talk about that. If you have questions for the exam, we have to talk about that. After the exam, I'll just show you this just to see where we're at. Here's the contents of our textbook, and here's what we've done. We did the first chapter, just introducing the topics of the course, and we jumped right to the 7th chapter because that's transcription regulation, and that's what those muscle differentiation videos are about. That's covered there too. And so we did those jumps, and then we went to part 4, and that's just a big part of our course: cell signaling, cell cycle, cell renewal, and cell death, like that. So cell renewal, stem cells, and cell death, and then cancer biology, like that. And we're covering these last 3 chapters on our exam on Thursday, and all the other material we've covered since this first exam. So, you know, the assignments, certainly, and, you know, everything that happened in class, and I've emphasized, you know, start out with the class material. That's a big reason why I try to record the lectures. Every lecture has a bunch of points from the exams. And you know, if they weren't on the first exam or the second exam, well, they'll probably be on the final, like that, you know, like that. So, you know, and there's just plenty of material to ask questions about to see how well you understand it. So after this, what we're going to do for the 3rd exam is we're going to go to section or part 3. And part 3 is what a person would call cell biology. And so what we've been doing, really, the first part of our course has been molecular biology. And the course is called Molecular and Cellular Biology. On the first day I described it. That's actually 2 topics. You could take a course at a really big university. You might still be able to take a course in cell biology and a separate course in molecular biology. Nowadays, they're almost always joined together, and the textbooks do it like that too. They're always a molecular and cell biology together, but they are, you know, potentially distinct topics like that. And section 3 I would call cell biology. And so after this, we're going to go to chapter 10, the nucleus. And the nucleus is a subcellular organelle. That's a big part of cell biology. Is what is it about the cell that we can't totally take apart down to the level of individual molecules? And the answer is, well, the cell itself with its plasma membrane is a super significant structure for regulating life, as are these various organelles in eukaryotes. And so if you look at the chapter topics here in this, it's all about, again, eukaryotic cells and the complexity of the cell biology and the organelles, and so the nucleus. What happens to the nucleus? Well, we've got a good start on that, but we'll add our examples to that chapter. So, for example, we famously already know FOXO doesn't get into the nucleus. Well, what does that mean? You know, it's blocked from getting in, but what's it like? What does it mean to get into the nucleus? And so that's what this chapter is about, and we'll bring our examples to that, you know, and build on that. And then we'll cover some other chapters like that. Some of these other chapters we're never going to touch on, and so I'm going to put some of that information also into these chapters, and we'll talk about it as we go, and we'll see how far we get. And so that's what we'll start talking about today, but we're not in a hurry because that material is not on this exam. So let's just start and talk about anything, questions about anything at all people want to talk about. Yep.
I'm a little bit confused on the regulation of stuff and the G to M transition.
Yep.
What's the difference?
Yep.
You said something about like the cyclins.
Yeah. Yeah. So this Dornique is asking about the cell cycle. And so we really need to be able to tell the difference between the G2 to M transition, the DNA damage checkpoint prior to entering mitosis, versus start, the G1 to S transition. And last year, a student met with me and asked me, like, they're really struggling to keep those apart. And I told them, I said, so, you know, in your home or your apartment, if you have a living room, you know, just put a circle out on the floor of the living room and have your couch be start and have a chair on the other side of the room be the G2 to M transition. Because they occur at different times in that time circle, and they're totally different stories. And we can physically separate them and think, oh, you're talking about S, you know, and entry into S, well, that's, so I only think about that when I'm sitting on my couch, like that. And I've described before how, you know, this is not a course in medical school, nothing like that. You know, I know these are biomedical topics, but it's still a college course. However, we can look ahead to folks who go to medical school, because they just have to, no matter how smart they are, they have to ramp their learning up, up. And they use tools like that, tools like I'm trying to introduce, where draw it out physically. Because, you know, ancestrally, ancient humans, right, they didn't draw, write, you know, but they lived in the world, like how you organize your kitchen and your, you know, space, right? You say, oh, the kitchen, that silverware is in there, right? And you can replicate that with book learning. You know, you can physically place it. So it's a different place like that, you know, and I kind of said, oh, stand up here for G2 to M, and I'm gonna go way back there for that space like that, because they're different, okay? And there's a couple of things they have in common. One is that the action proteins in both cases are CDKs. So in both cases, we want to know, what do the CDKs do? When we say we're going to go through either of those 2 points, what happens? And at the G2 to M transition, we learned that CDK1 is the one we need to learn there. We don't need to learn the numbers and names of the others. But CDK1 works with cyclin B at the G2 to M transition, and it's a DNA damage checkpoint. And the regulation has nothing to do with cyclin B. CDKs are cyclin-dependent kinases, but the regulation has nothing to do with cyclin B. It's accumulated during G2, and there's plenty of it. The key concept at the G2 to M transition is that there are some phosphates that have been added by kinases to that CDK1. Some of them are required activating phosphates, and we aren't learning about those, but 2 of them are inhibitory phosphates. And they need to be removed for the cell to go into M phase. And if they get removed, the cell will go into M phase. It's a very simple biochemical switch, just that simple. And that's why we went there to study that, to get a feeling for, you know, when we say, oh, you know, there's a red light, a green light, you know, a school patrol. Those are just, you know, real world analogies. But what's it really like is it's biochemistry. And how complicated is the biochemistry? Pretty simple at the G2 to M. So let's really embrace that. And what happens is just that phosphatase removes those inhibitory phosphates. And the phosphatase is CDC25. And if there's DNA damage, that phosphatase is going to get inhibited. We're not going to remove those inhibitory phosphates, and then we're just going to sit at the G2 to M transition until the DNA damage is corrected. And so upstream of the CDC25 is a kinase, CHEK1, that can add that phosphate to CDC25 and block it, stop it temporarily. And CHEK1 gets activated by certain types of DNA damage that occur frequently at the G2 to M transition. We learned a little bit, very briefly, that includes checking to make sure the DNA has been replicated. And I mentioned that in embryos, sometimes embryos are growing so fast that there is no G2 or even a G1. It just goes MS, MS, MS. So that's good to know. You know, when we think about the cell cycle, we think about, oh, just the typical mitotic cell cycle that occurs in our bodies, you know, as we walk around. But there are specialized versions of it in you know, various, you know, creatures, eukaryotes, animals, plants, and single-celled eukaryotes. And so that's part of the story also. but a minor part for us. And so then we want to know about, well, what is the action? And so we want to learn that CDK1 is going to cause the cell to be in mitosis, and it does that by phosphorylating a lot of very specific target molecules that make sense. It has so many target molecules that it has to activate some helper kinases, Polo and Aurora, and they all work together to phosphorylate these targets. And the targets can include, or do include, things that cause the chromosomes to condense and become visible. And that's a hallmark feature, a number one feature definition of mitosis, M phase, is you can see the chromosomes. In other times in the cell cycle, you can't. So scientists have seen and studied mitosis for 100 years because they can see it happening under the microscope. The chromosomes condense, they line up, they separate, the cells divide. And that's action by CDK1 and its helpers. And so the way those chromosomes condense is CDK1, Aurora, and Polo all phosphorylate targets. And for us, primarily histones, because we learned before, histones fold up. Histones are in nucleosomes and they fold up the chromatin, and they can fold it up further. The book, and we mentioned briefly in class, there are additional targets, condensin, cohesin. They're also phosphorylated to really shorten the chromosomes, really package them down so we can get them organized and get them separated. And that's one target. Then another target we learned about is that in lots of eukaryotes, not all, the nucleus actually disappears, disassembles and disappears temporarily during M phase, and then it reassembles, reappears. And the nucleus is, as we'll learn in chapter 10, a membrane-bound organelle, and those parts of the membrane, the phospholipids, actually just disassemble into little droplets scattered throughout the cytoplasm temporarily. The reason that they were together is primarily because of some proteins called nuclear lamins that are really like structural proteins, like you're building a house and you're going to put up some timber to have the shape of the house. And these nuclear lamins get phosphorylated, and when they're phosphorylated, they depolymerize. So they go from long, like timbers to individual monomers temporarily. They're still there, and but they are disassembled during mitosis because they get phosphorylated. So action, and there are lots of other actions that happen, and but those are our favorites because it's good to appreciate. That's the biochemistry of it. They don't get phosphorylated if there's DNA damage. We don't see the chromosomes condense if there's DNA damage. The nucleus doesn't disappear if there's nuclear damage. So that's G2. And there would be more to say about it. The book says more to say about it. There's certainly more to say about mitosis, but we're focused on just that transition, just the switch. And that's the core of it. And it's just different than the G1 to S transition start, the restriction point, which it takes place at a different time and could be considered a different physical space or time. And there, it's again cyclin-dependent kinases with different numbers that we don't need to know and different cyclins that we don't need to know. And there, phosphorylation is still an important regulatory step, but we decided that we wouldn't focus on that because we have such a good example of that over at the G2M transition. So to help make start seem distinct and different, we're just not talking about the phosphorylation of the CDKs and regulation of them at start. That happens, but it gets really complicated. And that's, we are saying that all the time. Start is the one we really care about because that's a control point. That's where the cell is really listening to external factors like growth factors like PDGF and so forth that are going to start the cell cycle. And we say the cells are waiting at a red light. And most of our cells spend most of their time waiting at a red light because that includes all of our brain cells that are really stopped, apparently permanently, in the cell cycle. And they're just sitting at that red light, and they seem like they're never going to enter it again. So scientists like to say that's a special detour called G0. But it's not. They're just sitting at the red light. And the focus of the question is about that red light and how it turns green. And before we describe the details of that, it's really important to make the point that in the next chapter it includes stem cells. And we find that Yamanaka Another scientist discovered that you could drive any one of the cells in your body through the cell cycle. You could make the light turn green. And you can do it with all the cells just by forcing the expression of MYC, a wild type MYC, that can make it happen. And so we'll talk a little bit about MYC, but just to be aware of that our cells spend most of their time in G1, whether they're living and doing gene expression and making insulin and so forth, or they're arrested at the end of G1, at that G1 to S transition, and waiting, just waiting like that. And they are just waiting like that. And without experimental intervention, there's no way for our brain cells to go back in, and no way for our differentiated muscle cells to go back in, or our kidneys. So it's a very interesting situation in our bodies and in animals and plants on Earth. So the nuts and bolts of it are that we wanted to focus on, well, what is it that regulates the CDKs at start? And a very common thing at start is there's not enough cyclin. And cyclin's a protein, so the proteins have to have genes, and so the target for MYC is expressing more cyclin. And I would say I think that's the only way MYC gets cells to go through G1 to S, is make more cyclins. The CDKs are cyclin-dependent kinases. There's not enough cyclin protein. What's it going to do about that? It's going to be a genomic response to PDGF to make more cyclins. And in some cases, MYC does that. In other cases, another transcription factor activates MYC. And a variety of transcription factors can activate cyclins, right? So that's one way to regulate start, to make the light turn green, is the rate limiting step in a lot of cells is not enough cyclin. So just make more and you're on your way. And we looked and should be able to draw on the test that just as a sort of general cyclin gene, our one drawing that has lots of enhancers, right? Because it's got the AP1, the June Foss enhancer, it's got the ALK SRF, it's got a beta-catenin, a TCF, it's got a MYC. I don't remember if we added others to that too, but it's just got a bunch of different enhancers, and there are a bunch of different cyclins. So when we draw that, we're kind of drawing a general idealized cyclin, saying there are many, let's just draw one and say it has all this regulation. But it might be D that has some and E that has others, yeah?
Does the beta-catenin bind to the TCF, or does it have its own separate?
Yeah, so beta-catenin always binds to the back of TCF, because beta-catenin is a transcription factor that's called a co-regulator. In the case of beta-catenin, it's a co-activator. And we learned about that concept for the first exam, and we want to just carry that through. So lots of transcription factors have a DNA binding domain, and they bind directly to the DNA, and we could make a list of those because it's most of them. Some transcription factors just don't bind directly to the DNA. They have to bind, like piggyback on another transcription factor. And so that's what's always going on with beta-catenin, is it does not have a DNA binding domain. It does not bind to the cis-regulatory region directly. Instead, it has to bind on the back, and it binds on the back of this TCF. And that's a really good example because TCF is always a repressor. So, in a lot of cells, they're not going to make cyclin because TCF is repressing it. Beta-catenin can overcome that repression and switch from a repressor to an activator. Remember that when we start our course, and we say most of the time scientists call those cis-regulatory regions enhancers because of the historical way they were discovered. The enhancer is simply tethered to the transcription unit, and can the leash, the length of distance from the enhancer to the transcription unit start site, promoter, and start site, can be variable distance, just like a dog leash. And so that was surprising to scientists, that the enhancer could be moved around, could be upstream the gene, downstream the gene, could be in an intron in the middle of the gene, and function just fine. And it's because the enhancer is serving to create increase in local molar concentration, because it's just tethering or leashing that transcription factors like MyoD to that location. And so, enhancer is not a great word. So lots of scientists would like to call those enhancers response elements because they're part of the cis regulatory region and it's a response element. And we first learned that for our first exam and our final with the estrogen receptor, nuclear receptor, because in the absence of the hormone, they're repressors, and in the presence, they're activators. So that doesn't make enhancer a good term for an estrogen response element like that. So the same is true with beta-catenin target genes, is the enhancer is really a response element because it doesn't just enhance. In the absence of beta-catenin, it's repressing like that, yeah? And so, yeah, I would say beta-catenin always binds on the back of TCF. It's interesting, and we don't know the answer in our course, but like if there's no beta-catenin and TCF is repressing cyclin gene expression, can MYC overcome that TCF repression? And I don't know. The answer is probably it's a case-by-case basis like that. But things get complicated like that. The enhancer usually has the cis-regulatory region for lots of genes, and certainly these cyclins has a lot of different opportunities for transcription factors to bind and activate or repress. And we just got a little taste of that. A key thing about that I mentioned once and mentioned again is that the bottom line for any transcription factor is what is the expression of the target gene? You know, where, when, and how much it's made. And it's kind of like the cis-regulatory region works like a kind of majority rules. You know, you can have some repressors, some activators, and well, what's happening? What's the output like? You know, and so scientists can struggle with the variety of, transcription factors that are regulating the gene, or they can just look at the RNA levels of the gene expressed in the cell, because that's an indication that, oh, the gene is being expressed, you know, at this level, like that, yep. So that's one, right, is that at start, the cyclins can be rate limiting, and they often are. And all we need to do is make more cyclins, and we do that by expressing. So in fact, we could start with a wound and say, start with a wound and show how those skin cells get activated. And that would be a long drawing, because you'd start and you say, well, the platelets release PDGF. PDGF is in the extracellular fluids outside of the skin cells under a scab. It binds the PDGF receptor that is always present on every one of our skin cells because they're prepared to be at the site of a wound for our whole life. And it activates the PDGF receptor, so I don't know, phosphorylates, so you know more stuff around RAS, RAF, MEC, phosphorylation of ELK. ELK is going to activate stuff. And we learned for our first exam, well, ELK could activate MYC. It could activate a variety of target genes. And maybe MYC activates cyclin. But there are a variety of target genes, but what we're talking about right now is really typically the story is for PDGF is going to be, it's going to activate cyclin gene expression. We're going to get enough cyclin protein made to go through stuff. And you know, there you go. The skin cells are off to the races trying to heal the wound like that. We learned a little bit that under a wound, there's a bunch of different things, right? That SRF that also is regulated, that actually is considered a more significant regulator of wound healing than is ELK and the PDGF receptor pathway. And then we learned about other transcription factors that also get activated under a wound, right? Because sometimes there's scarring, right? The wound is so severe, it goes down into the dermis, not just the superficial epidermis. And you know, you can have a variety of wounds like that. And the scarring is done, you know, to try to create a healthy recovery from a wound, which would be very significant and, you know, a very important for a natural selection and the evolution of wound healing. To, you know, get that primitive ancestors of ours back onto their feet or their fins, right? Because, you know, lots of vertebrates have to go through wound healing, and it's always the same stuff like that. So that would be a big part of it. Then the other thing that we want to bring up in the G1 to S transition is that unlike G2 to M, there's also the story of these small inhibitory proteins. And I don't know, but I think most scientists call them that. Our book likes to use some other terms. That's a nice word because they're inhibitory. and they're small proteins. And their smallness is indicated by their size. They're surprisingly small proteins. And so P21, P27 are the ones we learned about, you know, that are especially important. We're not gonna bring up other ones of those on the exam, even though they're on a table in our book like that, you know? And so we learned good stories about P21 and P27. And P27 was just a very quick story, which is that P27 actually is a thing where it's gonna try to inhibit the start, and it won't let go until the light starts to flicker green. And that means that some CDK molecules have been activated, and they actually, in their targets, include P27. So P27 gets phosphorylated and destroyed, and that causes the light to turn fully green. And that's one of the complexities of start, is that we have to make a big dramatic switch. You know, we gotta go from a red light to a fully green light that all the cells can see as fully green, and they'll move forward like that. And a lot of times it starts out kind of flickering and then gets fully green. So the P27 is just one little example of that, to be mindful of that reality. And then the P21 is the one we spend time on, because that's a thing where this is a G1 to S control point. The cell's really looking for external signaling to activate it. However, in part of that is also a very different DNA damage checkpoint. And that makes sense, and it's good for us to be able to explain that. You know, you don't want to go through G2 to M transition and have a cell divide while it still has damaged DNA or not fully replicated DNA. Similarly, you don't want a cell to go through S phase and replicate damaged DNA. That would just, you know, permanently, you know, cause trouble, because you can't fix it now, you know? So there's a big, careful DNA damage checkpoint. And that's the one that P53, the guardian of the genome, is controlling. And P53 normally gets degraded all the time. And when there's DNA damage, it gets stabilized and it starts getting phosphorylated. And initially it gets phosphorylated, and that causes it to bind to the P21 cis regulatory region, and we start to detect expression of P21. And we get P21 RNA, of course, and then P21 protein, and it just is a school patrol officer that goes, I don't care if the light is green, you're staying stopped, like that. And so that's why P53 is the guardian of the genome, like that. And we saw, just as a side note, right, that HPV virus can cause the destruction of P53, right? And also retinoblastoma, right? Those viruses are just trying to get the cell to turn into a replication factory, right? And so that's an important way to further appreciate the significance and sort of the danger of that G1 to S transition. And with that DNA damage, we get that transition to the next chapter, from the chapter 17 of the cell cycle into the cell death chapter in apoptosis, because amazingly, the protein that phosphorylates P53 will hyperphosphorylate P53 over time, and hyperphosphorylated P53 picks up another target, and that's that P21 gene. So we have some examples of the different types of regulation, and you know, it gets, I think, confusing when we add the two DNA damage checkpoints like that. But that's, you know, just you know, a little bit. And then I think, you know, the start is just, there's more to say about it, but we're trying to get a good introduction to the players, you know, the most famous players like that. You know, I can't remember right now, but is P53 mutated in like half of human cancers, you know? And that's because, not because it's gonna cause cancer, but cancers just keep building up more and more random mutations, and some of those will cause further trouble. And the cancer will be more aggressive, you know, and spread or grow faster, you know, like that, cause more trouble. And it would be helpful, you know, for a cancer if it's not sitting there, because P53 is trying to guard the genome from further damage. So in the case of cancers, apparently it's pretty you know, effective for P53 to get mutated and lost and lose that DNA damage checkpoint, and just, you know, just have the cells roll through start like that. Yep. Yeah. So it's a big dose of stuff, right? But it's just like, you know, like that guarding the genome. Every time I say it, I choke up, because it's just such a cool, you know, biochemical fact, of what it's up to, you know, and, you know, there should be Marvel, should be in the Marvel universe where they're like, yeah, you know, here's a movie about B-53, the superhero, like that, you know, that would help us, right, help your learning. It's like, dang it, why didn't somebody make a cool summer blockbuster about the cell cycle? And just the various players of the cell cycle sitting around eating shawarma or whatever they do. Yeah, okay, and so, yeah, so, yep, yeah, and part of this, right, you know, is that, you know, Dornika's question was scattered throughout several class meetings and in our book and stuff. And a big part of biology, and, you know, is for you to pull it together, you know, and draw it out like that, you know, include that HPV virus, right, because that fits into your drawing too, you know, like that, so, yeah. Sometime I'll show you, I had a cell cycle assignment once, and a student built a cell cycle tower like this, where it was different layers of the same thing, but focused on different aspects of the cell cycle. Yeah, I'm trying to remember to post that, because you'll just be like, oh my gosh, right? Because we're not artists, you know, I'm not, I'm left-handed, you've seen my writing, you know, my projects and assignments are not pretty. But some students are, and you're just like, oh my gosh, and you know, you're not getting extra credit, but wow, you spent about 100 hours on that cell cycle assignment. Yeah, because you made a tower. Yeah, so that's that. Yeah, and more on that or other things that, you know, we need to know for our second exam. Yeah, Chase?
Yeah, this is a small thing. It was from one of the assignments. It had to do with MCM proteins, the mini chromosome maintenance proteins.
Yeah.
And I know they're a DNA helicase and they're necessary for like transcription and or
Yeah.
not transcription or DNA replication.
DNA replication, yeah.
And I'm just curious, like why I'm not really sure why like a mini chromosome as like was in the video was needed to discover it, I guess. Does that make sense?
Yeah. Well, so two things. One is, that's a good question to ask right away, because I didn't actually remember to talk about that. So at both the G2 to M transition and the G1 to S transition, it's nice to talk about, well, what do these action CDKs do? And we learned, well, they phosphorylate targets at G2 to M, and we learned a couple of those targets, and you're like, oh, okay. You know, it should have just a permanent shift in your understanding of what it's like to be a cell. You know, they're going through these steps and regulated by, you know, clear things that scientists know a lot about. So I didn't mention that when we were talking about the G1 to S transition. Is when the cyclins when the CDKs at G1 to S get activated, what do? Well, they're kinases. They add phosphates to targets, and we learned about some of their targets like that. And and so MCM gets phosphorylated, and those are these proteins that bind at the origins of replication. And the book has a fairly complicated figure of what's happening at origins of replication. And origins of replication are where the DNA replication begins during S phase. Chromosomes are so long, millions and millions of nucleotides long, there need to be many origins of replication. Each origin of replication looks the same. They're just a little place to get the DNA polymerases to start and get going, like that. And to help the DNA polymerases to find the place to start and get going, there are these proteins that assemble. And for us, MCM is great. Two things about it that we learned. One is that it gets phosphorylated. So we gotta get that phosphorylated to get it active. The other is, we're gonna need more. And so the MCM is a protein that must have a gene, MCM gene, and that's gonna have to get increased. So that's also a target of these transcription factors that are gonna activate START, is they're gonna try to push up the expression of all the proteins. And that's a good example of that. And you know, what kind of transcription factors activate all of the genes that need to be used during S phase? And we talked about a little list of examples, and one would be the MCM gene must have an enhancer that gets activated at the start, and it gets activated by this family of transcription factors called E2F. And E2F binds to many genes. Another example would be DNA polymerase. There's some DNA polymerase always around to help with DNA repair. But during S phase, we need massive amounts of DNA polymerase because we have to get it to work at every origin of replication. So the DNA polymerase gene has to have a great increase in its expression. And these E2F proteins are transcription factors that are always bound to the cis regulatory region of MCMs and DNA polymerases, and then other genes that make sense. For example, we're going to have to synthesize more of the AGCT nucleotide precursors. We have to build those. And enzymes in metabolic pathways build those. And those enzymes are proteins, and they have genes, and we need to activate and increase the expression of those enzymes. Yeah? And a good way to appreciate that is back to that HPV virus, and other viruses that are like, oh yeah, you know, the E2F transcription factor family is like an Achilles heel in a eukaryotic cell. It's in charge of massive amounts of DNA synthesis factory stuff. And if that HPV could get the E2F going, it would get lots of help in copying its DNA to make more viruses like that. And the E2Fs are always there on the cis regulatory regions, bound to enhancers of hundreds of those genes that are really expressed at high levels during S. And that level of expression activity is the definition of S. You know, if you're making a lot of nucleotides and DNA polymerases are using a lot of nucleotides, you know, copying a lot of DNA, you must be in stark, you know. So E2F is that. And it's say Achilles heel because it's like a vulnerable spot. If HBV could get those going, and how does it do it? It just destroys retinoblastoma. It decreases activity by, you know, gets phosphorylated, and that role of retinoblastoma is it's considered a tumor suppressor. It's like stopping entry into S phase by just binding to the backs of all those E2F. So back to that question that Spencer asked. So those E2Fs, those must be transcription factors that have DNA binding domains. And those enhancers for all of those S-phase, or many S-phase genes, they must have binding sites, enhancers that bind E2F family members. And then retinoblastoma. Retinoblastoma is one of these co-regulators. It does not have a DNA binding domain. It binds on the backs. It piggybacks on the back of the E2Fs, and it represses them. So the E2Fs are there, they're ready to go, and retinoblastoma is stopping them. And if we get rid of that, we can. And it's another one of these flickering green light things where in a normal healthy cell, the number one way that retinoblastoma gets released and the E2F gets to do its job is the retinoblastoma gets phosphorylated by CDKs. So that's another action item, right? Is, okay, the CDKs at start are going to phosphorylated MCMs like that. Another thing they're going to do is they're going to phosphorylate retinoblastoma. And that's it's turning the right, like green. It's leaning into it, right? Because you're like, okay, well, we're starting to make a lot of DNA polymerase and it's ready, you know, like that, and making more MCM, you know? And there's checks and balances there, so it means like it's leaning in, it's flickering green, you know, we'll need some other things to go to make it fully go. But it's, yeah, it's really leaning into S phase. When you're making all the components to be in S phase, you're pretty close to being in S phase, like that. So another action item, yeah?
Can we review the drawing for the origins of replication?
Yeah, that actually gets back to Chase's actual question of like, well, what's the deal with that? Uh-oh. So now, yeah, okay. So, yeah, so for this origins of replication story, for our course, you know, we're going to kind of keep it pretty simple. So there's DNA, a stretch of DNA, and that's a chromosome, and we're really zoomed out, millions of nucleotides long, and the DNA sequence, the AGCT letters, establish locations like this, and they're kind of they are you know, distributed unevenly like that, and those, each of those is called an ORI, and that's just an origin of replication. So they're all equivalent. You see the same thing happening at each one. And so we could erase that and just zoom in on one ORI, like that, one origin of replication, and it would look, essentially the same in each case. And so now, we're zoomed in and we're just looking at this. And for us, the drawing is much simpler than one in the book. For us, we would want to be able to say that, well, that's got to be where DNA polymerase shows up. So we want to be able to add that. And then there are a whole bunch of different proteins that are going to help. And there's a good chance that you ran into some of them in introbio or genetics. For example, there are helicases and topoisomerases. We've got to unravel this DNA. And we didn't emphasize that, but just to be aware that the drawing is more complicated. For us, the favorite thing is that there are these MCM proteins that bind in this location, and they're a type of helicase, so they're going to help with a function. But for us, it's just, we don't really need to know one. For us, it's just especially that they're playing a role at the origin of replication, and that to do their job, they get phosphorylated to really be able to get them there. And it's a significant thing, right? Because we don't see many examples of this in our course, but these are DNA binding proteins. They would have a DNA binding domain. They would bind to a specific DNA sequence, but they're not transcription factors. They are involved in DNA replication. And we haven't really dove into DNA replication, so it's not too hard for our course to distinguish between DNA replication and RNA transcription. So DNA replication is what we're talking during S phase, copying the DNA. And so the drawing would be pretty simple about like that. And I wouldn't say, gosh, I was hoping that you found more out of the book. When I ask questions, if I ask questions that are like from the book that we didn't talk about, the answers I always say are very unlikely to require you to make a more complicated drawing. Because the book has lots and lots of figures with lots of components, and that would be pretty tough, to say I could ask you to draw all the details of, that pathway. And on the other hand, instead, I would try to ask questions about, something that's explained in a section, maybe a method, the whole page, or a bold term that's not just isolated, it's used in a whole thing. Yeah?
Sorry, what is the blue circle?
The blue circle is unlabeled because it's just an indication that there are a whole bunch of other proteins, and it could be a helicase, and there are other proteins, but just to show you that, because the question was, let's take a look at that drawing. And for us, the drawing is kind of obvious. Yeah, it was going to have DNA polymerase molecules showing up like that. But for us, the regulators MCM1 are ones that we really focused on. And then to loop all the way back to Chase's question, I'll just, so I just described briefly the experiments that were done to identify the MCM protein. And in the field of molecular biology is a joined together of genetics and biochemistry. And so the work always involves stories of biochemistry research or genetic research or combination of the two, and that's a story for MCM. And so the scientists had no idea how origins of replication were regulated. Who has to bind there to get the job done, like that? And so they said we should maybe pursue a biochemical strategy. We should maybe go to S phase and purify proteins that are bound to the DNA and see who's bound there. That's the biochemist's approach, right? This MCM was identified by geneticists. Take a genetics approach. Let's try to look for mutants that fail to have a effective functional origin of replication. And I just briefly described that. The way they did that is in yeast. So yeast are very simple single-celled eukaryotes. Every eukaryote has massive amounts of orthologs, including all of these, to get DNA replication done. So no surprises, they could discover the regulators of the origins of replication in the yeast that causes bread to rise. And the way they did that is, you know, you're like, well, for the yeast to stay alive, it's got to be able to copy its DNA, right? So we got to come up with some trick. And they came up with a trick to look at just a little part of a chromosome. So they made an artificial chromosome they called a mini chromosome. And they just put a gene on it, like, for example, GFP. And if it, you know, the mini chromosome got copied, the cells would glow. They would keep that, you know, the gene in the mini chromosome. And if they didn't, they'd lose the glow. So they had a screen, you know, to find mutants that failed to replicate that mini chromosome like that. And that's why it's called mini chromosome maintenance. It's just one of the genes that came out of that screen strategy. I'll just mention that when I was a first year graduate student, I took a course called Inside the Nucleus, and it was taught by a professor, John Lewis, who went on to be my advisor. And it was really the first time John had ever taught a course, and he's not a good teacher, he's a very good researcher. And John refused to ever talk about like a drawing of what was happening at the origin of replication, and instead would only talk about experiments that gave evidence of what was happening at origin of replication. And that was just brutal, like that. You know, to never say nucleosome and just say, well, you can see that the DNA is protected from degradation in a spaced way, like that. And so molecular biologists love to talk about the data rather than the drawing, you know, like that. John would say every model is false. Gives you a false understanding because it oversimplifies things, over implies things that are not necessarily known. Oh my gosh. And that course meant once a week from like 7 till 8, no, not 7 till 8, till like 10:30 at night. Yeah. And it was graduate students, and then some undergrads took it, and the undergrads would just be drinking whiskey and stuff, because they'd just be like, this is so messed up. Because John talked in a really ponderous way, and he hadn't taught before. Last thing I'd say about that, just because we have a test coming up, John thought it was cool to give exams. He was a researcher and did not want to teach classes. His exams were just, each exam was just 10 multiple choice questions. So you got one wrong, you know, 2 wrong, you know, 90, 80, 70, 60, right down the line, like that. Yeah? And his multiple choice questions would have not just like A, B, C, D answers, but like A through M options. Yeah. And then the answers would do things, because they were all methods, and they would just be a series of numbers. Like the answer A is 1, 2, 6, 7, never 9, 11, 15, 20. And over here is a list of what the number of things were. It's just absolutely brutal. Our exam's not like that. You know, we're just dancing along going, well, I mentioned mini chromosome maintenance because it's just a cute thing. It's such a great name, you know? And then we roll on, you know? So anyway, sorry. Yeah, it could be worse though, you know? You could be struggling at night taking an exam in the dark. Yeah. Other things people want to talk about? Anything at all is great. Dornika has one.
Can you please explain the big difference between all the 3 stem cells?
The big difference between all 3 stem cells. Yes, let's talk about that. So, stem cell biology. And what we want to say about stem cell biology is that stem cells are important to talk about because they're in the news so much that outside of class, you and everybody else has an idea what a stem cell is. And a lot of times that is what scientists call a misconception, you know, because your brain kind of filled in gaps. You know, you heard a little bit about some stem cell news, and you kind of subconsciously fill in gaps to try to make sense of it, like that. But the stem cell is just based on the way the cells are dividing. And they're like a stem where they'll divide like this, and each time they divide, they make a stem cell again, and then typically they're dividing asymmetrically, and each time they divide, they make a precursor. And our favorite stem cell from the beginning of our course is that muscle satellite stem cell. And it's an example of one of the three categories of stem cells. It's an example of what are called, and it's their second category of stem cells. These are called adult stem cells. And that's a misnomer, not accurate, because adult stem cells would be found in an embryo, a fetus, a child, a teen, and an adult. And they would include muscle satellite stem cells. They would also include those stem cells we talked about in the bone marrow. And those stem cell populations that are adult stem cells are maintained constantly for the life of the organism because they're said to be self-renewing. So see, when the satellite stem cells divide, we don't lose the number ever, because they're dividing asymmetrically and they're just making a stem cell again. And that's why they're called stem cells, because it's kind of like a stem, like at the base, and they're just making and maintaining that population. So it's said to be self-renewing, constant throughout your body, life. Imagine the bone marrow stem cells, they're making so many cells your whole life, but the population is not decreasing because they're dividing asymmetrically, and they make these precursors. And the precursors could be precursors that are going to be myoblasts to make muscles, or they could be precursors that are going to make the blood cells or other parts of our body, you know, variety of different stem cells that are adult stem cells like that. So that's a great example for that second category of stem cells like that. And then that's where that Yamanaka's research could really be understood. And Yamanaka is a scientist that said, oh, if we could find out what this stem cell is expressing to maintain its stem cell identity, that's the term they use, stem cell identity gene, well, maybe we could express that in a cell and turn it into a stem cell, and that's what they did. They found these cells are making specific genes, and only a small number of them that keep them being a stem cell. They did that by comparing it with these cells. These cells are they're stopping their stem cell status. They would be reducing and turning off stem cell identity genes because they're done being self-renewing, and they're going down a path toward ultimately differentiation. And so that's adult stem cells like that. Okay, and then the next category of stem cells that we should talk about is our first, and those are called embryonic stem cells. So of the three that Dornique asked about, embryonic stem cells is the first one that people like to talk about. And embryonic stem cells, it's kind of surprising to hear this, but they're only found in mammals. There's no chance they exist in other organisms. And you know, when we say that in biology, maybe somebody will find some someday, but they're not going to. Because other organisms that are not mammals make eggs. The females make eggs that are big eggs that have a lot of molecules in them that mom loads in. And lots of times those are, of course, nutrients that mom's providing to the early embryo because mom's going to lay an egg like that, release the egg. Mammals have this internal fertilization and internal development, and so the eggs are tiny. And so they lack those nutrients, but they also lack helper factors to help development. And so early on, as those fertilized cells develop in a mammal, they all are equivalent. They all could make the whole organism. And people refer to that early embryo as a pre-implantation embryo, because the early embryo is going to go down, the fertilized egg is going to continue traveling down the fallopian tube in a mammal, and then implant in the uterine wall and continue development. And in humans, that pre-implantation embryo is about about a week to 10 days. Fertilization takes place slightly different times, about a week to 10 days, and that's the only time there are embryonic stem cells. And it's just a special term for all the cells of a pre-implantation embryo. And we can see their potential by the existence of identical twins. So a pre-implantation embryo can split into two and have two identical twins form and grow and be totally normal. That just does not happen in a non-mammalian animal. There's no such things as identical twins like that. You could create one artificially through some scientific experimental trick, but not in nature like that. Yep. And again, that's a pretty bold claim, but yeah, I think it's pretty accurate. So they're just embryonic stem cells early, and they can make anything like that. We're not going to be expected to say too much about them, but we could say a couple of things. One is they're going through the cell cycle, right? Stem cells go through the cell cycle. And another is, if you think about it, one of the things that these guys must have is they must have a lot of unmethylated DNA. Those cells need to be ready to express all the genes. They have all the genes because of genomic equivalence, and they need to be ready to express them all. And once implantation occurs, the embryo is starting to already go down these pathways where some cells are already starting to head toward being specific cell types. Just big categories. If you happen to know the terms ectoderm, mesoderm, and endoderm, they start to decide, start to move. And that leads to some very early patterns of DNA methylation. They've already decided, yeah, I'm not going to be going to that brain pathway. I'm going to just be the endoderm and line the gut. So I can start to methylate some DNA. So it's just kind of roughly coincidental with that time of implantation where the cells have developed to the point where they're starting to say, yeah, no, I'm just not going to be able to make every cell like that. And they just move further and further down that pathway. Yeah. Yeah, so that's our first example. And those are the types of naturally occurring stem cells. Embryonic stem cells only found in mammals, adult stem cells found in many, many organisms. And then the third one are these experimentally produced transgenic cells that are the induced pluripotent stem cells. Like that. And we could talk about them, but it might be good to kind of pause there because they're a different story, right? And so it kind of helps separate your learning. We talked about the naturally occurring ones. We have good examples. Our favorite is the muscle satellite stem cells for the adult stem cells, and then these embryonic stem cells. And these induced pluripotent stem cells are made by forcing expression of as few as 4 genes, and what we want to know about them is MYC is one of them. All 4 of them are transcription factors. That's satisfying to hear, right? If you want to turn a cell into a stem cell, you're going to have to turn on a lot of gene expression like that. And the 4 genes include MYC to get the cell back in the cell cycle. And then the others are transcription factors that are going to help activate decreases in DNA methylation. Because remember, we learned in the muscle differentiation videos that when cells differentiate, like when a chondroblast cartilage precursor becomes a chondrocyte, a differentiated cartilage cell, it just starts methylating a lot of genes it's never going to use again. And if we want to take that chondrocyte and turn it into an induced pluripotent stem cell, we are definitely going to have to remove a lot of those DNA methylating marks on those genes like that. And so that's a good thing for us to appreciate, right? Is that kind of the 3 pieces of the induced pluripotent stem cells is, well, the scientists realized that those stem cells must be expressing some stem cell identity genes. To maintain their status, to be self-renewing, they must maintain expression of some genes that are keeping them going. And once a cell stops, how could you make it go again? And the answer is, well, based on what we know in our course, you've got to get it back in the cell cycle. Turns out that's pretty easy to do, but obviously a really dangerous game. You know, none of these induced pluripotent stem cells have been put back into a human yet, because they're moving through the cell cycle through, you know, forced activation. That's dangerous, right? That's kind of a pre-cancerous story. And then the other thing is, well, they're going to have to remove a lot of the DNA methylation, you know, because they probably have methylated lots of genes, and they have all of them. Genomic equivalence is there, but they aren't capable of expressing them all because lots have been methylated. Yep. So that's a good lineup of those guys. Yeah. Other things you want to talk about? Doing great. Oh yeah, we have 10 minutes left. Yep. Bryce has a question.
Yeah, could we go over BCL2?
Yeah, so BCL2 comes up because in the program cell death story, and it's in the apoptosis, so we learned a little bit about autophagy also, and they're regulated by different proteins. So BCL2 is regulating apoptosis, and it's the most famous anti-apoptotic regulator. So in apoptosis, there are these action proteins, and there's a family of proteins called caspases. They have numbers to identify the different caspases, and we don't need to know the numbers. They play slightly different roles. The book uses the numbers because they are important, you know, in a more advanced setting. And so in apoptosis, we wanted to learn a little bit about the action. You know, what's the action? So we learned about that. And what do those caspases do? They're proteases. They clip target proteins, and in clipping them, they don't degrade them. They just clip them once, and they remove part of the protein so that protein can be activated. Scramblase and so forth, like that, you know? And then that's the action, and then the emphasis is on in every eukaryotic cell, every plant and animal that you'll ever see. Those caspases are there all the time. It's kind of scary to think about, but every one of your cells is like, you know, thinking about going through apoptosis. I really am. And I think that's considered to be just part of the evolution of multicellularity, you know? Because it's still, you know, the basic building block of life on Earth is the cell. And it was a big deal to take, you know, our single-celled, our unicellular ancestors and say, hey, you want to team up? You want to make a team? And they were like, F no, I'm on my own. I'm a loner, you know, like that. Yeah, and so part of that collaboration is, hey, you know what? You can be on the team, but you are going to be concerned if you start getting out of line, because you have that kind of rowdiness from your ancestry of being a single-celled organism, and we have to kind of slow you down and tame you, and we're just going to ask you to be prepared to kill yourself whenever it's better for the organism. And you're like, oh man, I don't want to sign that contract, you know, like that. But I really think that that's what's going on, is that it's going to be, you're going to have to express those caspases constantly, you know, because you're on a team now, like that, right? And therefore, we know what they do to action, and then they must have really important upstream regulation, and they do. And the only family member of the BCL2 family member that we want to name is BCL2. And it's an anti-apoptotic factor like that. So it always is trying to repress the activation of the caspases. And it's in our cells right now. And then we, you know, took a gentle step into that family. The BCL2 family has maybe about 15 or so members in humans. Some are pro-apoptotic, some are anti-apoptotic, you know, and so we need to know that. We just you don't need to know the names of them. We just need to know the names of BCL2. And the BCL2 and caspases were really originally discovered in nematodes down at MIT in Boston using a big genetic screen. So, you know, molecular biologists that were also doing a lot of genetic work. And they had funny names, CED, SED3, and so forth like that. That's not a name we need to know. We need to know BCL2. And it's just an upstream inhibitor like that. And we didn't learn about its specific mechanism of action, of how is it inhibiting the catabases, but just that it's an inhibitor. And then the next thing we learned is that, our book puts the cell death chapter in the cancer section of the book right after the cell cycle, because cancer is understood to be cells growing out of control by going through the cell cycle too much and then failing to go through apoptosis. So that's a big check and balance of the cell cycle, is just about any time a cell is stimulated to turn the light green in that same cells, it's activating members of the BCL2 gene family to either lean into maybe, this is, like going through START should be scary. In a multicellular organism, for a cell to go through START every time in the bone marrow, the cells should be scared, because they're activating freaking MYC, and MYC is cranking up the cyclin, and you're like, geez, I don't know. And the cell should be careful and be ready internally to just say, yeah, this is not good. Yep. And so that's shown in the figures all throughout our book like that. There'll be examples where it's showing here's the growth pathway, and then it's like it's also regulating these pro-apoptotic and anti-apoptotic vectors. And so we just learned one example of that, and that's this B-cell lymphoma. BCL2 stands for B-cell lymphoma 2. B cells are white blood cells in our body that make these antibody proteins. And we just learned one example that we need to be able to draw on our exam, like we did on the first exam, an example of a random translocation between two genes. And the story is always the same. We're going to accidentally repair a translocation between two chromosomes by accidentally connecting the transcription unit, in this case of BCL2, to a cis-regulatory region. And we're going to get a misexpression of that gene. And in the BCL2 story, we're going to connect it to the antibody gene, cis-regulatory regions. There are 3 different antibody genes. We don't need to worry about that. Just we get a translocation. B cells express a lot of antibody. They're now going to express a lot of BCL2. And we get that low-grade non-Hodgkin's lymphoma out of that. If that's the first step, what happens is those blood cells are supposed to normally die after a couple of months. We're making new blood cells all the time because we're continuously turning them over, making our immune system brand new over and over again. And those cells should die. But if they overexpress BCL2, it's an anti-apoptotic vector, and they just don't die. And so that's why it's called low grade. And the doctors can see this patient and they could be, wow, you just have way too many B cells. I don't know what the symptom is. I'll guess they're tired. They're just swamped. Their red blood cells are kind of swamped out by these way too many B cells. And so somebody that has low grade non-Hodgkin's lymphoma is, they don't have cancer, but if they get a mutation in an oncogene and they start making too many B cells, they're in real trouble, because the normal B cells are already failing to die, and now you're making even more of them. And so that's just a dramatic example for us to appreciate the role of BCL2 and how it could get disrupted. And there are lots of other examples of that, about cell death getting disrupted like that. But what a memorable one to learn about. And a further example of what we're kind of diving into in that part of our this part of our course, which is that each specific cancer person can get will have just a specific molecular mechanisms that are a problem, and that's the case with these people who have this BCL2 lymphoma like that. Yeah, and we're running out of time, so we got to wrap it up. But you can send me questions. We can plan to meet. When we get here, 9:30 on Thursday, we'll set it up. I said, we have to have assigned seats, but it seems like we have enough students in our class now. We can spread out. So come in and put a space between you and any neighbor, and we can move desks around and stuff like that. So you're comfortable, but also sitting, a space from other students. And right at 9:30, we'll get started and plow through this second exam. And our recording works.